Bismillahirrahmanirrahim. Now we have, as I've been requested by some of you to do this video. This contains all the syllabus update on the new topics which have been added to the 2022 syllabus in which you're taking the exam. So all the new topics will be covered in this. First we're going to cover the paper 2 topics, and then we're going to cover the paper 4 topics. So this is continues only the paper 2 topics, which is if students are taking their AS biology exam, then they need to go through this very thoroughly. In fact, they should go through it once or twice so that they can remember everything which has been updated. The first thing to remember is that your paper 1 now is for 1 hour 15 minutes. This was not for 1 hour 15 minutes, it was only for 1 hour in the previous exams which were held till 2021. So I think the difficulty level is going to increase. So the paper 1 now is for 1 hour 15 minutes. The paper 2 is for the same time, 1 hour 15 minutes and 60 marks, and this was also 40 MCQs were in that. Paper 3 is still going to be the same 2 hours and it's going to be a practical exam. So nothing else has changed. 2 hours, paper 3, practical exam, which some of you might have taken an exemption. Well, if that's the case, I don't agree with that, but then it's your choice. Now the AS level has remained the same 11 chapters that we have, and these 11 chapters are the same which was in the previous syllabus, but individually some things have changed. Otherwise the 11 chapters are the same as the previous ones in the previous syllabuses. So 11 chapters: cell structure, biological molecule, enzymes, cell membranes and transport, mitotic cell cycle, nucleic acid and protein synthesis, transport plants, mammals, gas exchange, infectious disease and immunity. But many things have been removed, and I'm going to just give you a list of all what has been removed from the syllabus. The following have been removed from the syllabus, so please do not do the MCQs on this or the questions on this, because you tend to keep on asking me stuff and then I have to tell you this is no longer in the syllabus. So leukemia, myasthenia gravis, autoimmune diseases, smallpox, measles, high altitude and RBC count, rate of transpiration, smoking, lung cancer, COPD, effects of nicotine and carbon dioxide, all have been removed from the syllabus. Please pause the video here and please remember all this is not in the syllabus anymore. As you can see in this part, I'm just going to be viewing the points which are new in the syllabus. I've made a whole a study on this and spent a lot of time in finding out what is new in the new syllabus. So Cilia microvilli are new in the syllabus. Compare the structure of a typical plant and animal cell is new in the syllabus. State that cells use ATP from respiration for energy requiring processes. So that is a little new in the AS level, because this was previously a little more referred in the A2 level. And the fact that some viruses have an outer envelope made of phospholipids. So these are the 4 new things which I'm just going to discuss with you all, and then we will proceed on to the next chapter. Microvilli. Now microvilli, as you know, has been added lately to the syllabus. Now when I look at it as is infoldings of the cell membrane. So it's actually in the cell, in one cell. This is one cell. And in the cell, these are called microvilli. So what are microvilli? Singular microvillus, small finger-like extensions of a cell which increase the surface area of the cell for more efficient, number 1, absorption or secretion. Now something has to be thrown out of the cell, or maybe something has to be absorbed into the cell. So for absorption and secretion, and I'll just show you another diagram which also shows this. Of course, is my diagram. Now here you can see the microvilli which has been labeled here. This is from the new book which I've taken this diagram. So this is the microvilli which I want you to see is, and how this can be given to you in the exam, and maybe an MCQ on it or something like that. So microvilli. As you look into the structure of the centrioles and the centrosome which is expected of you, the extra resolution of the electron microscope reveals that just outside the nucleus of an animal cell there are really 2 centrioles, and not one as it appears under the light microscope. Now, triplet of microtubules. So you look at it, there's a 9-3 structure. There are 9 triplets. And the centriole is one of 2 small cylindrical structures made from microtubules found just outside the nucleus in a region known. Centrosome is the area which is outside it. So centrosome and centriole, please understand the difference between the two. And this is a 9-3 structure. You see, each one is 3, 1, 2, 3. And there are 9 of these. And a centriole, hollow cylinder about 500 nanometers long, and is formed from a ring of short microtubules, and each centriole contains 9 triplets of microtubules, figure 1.30. This is from the new book, which I hope you're studying the new edition to the biology, which has been now for 2022. There's a new book, and everybody should be following that book. The centrioles acted as microtubule organizing centers. MTOC is for microtubule organizing centers for the assembly of the microtubules that make up the spindle during nuclear division. It is now known that this is done by the centrosome, but does not involve the centrioles. However, centrioles are needed for the production of cilia. Centrioles are found at the bases of cilia and flagella, where they are known as basal bodies. And the microtubules extend from the basal bodies into the cilia and flagella and are essential for beating. So, centriole structure you must know is the 9-3 structure. Now, coming to cilia and flagella are identical, they are identical structures, and they're whip-like beating extensions of many eukaryotic cells. And each is surrounded by an extension of the cell membrane. You must understand that there's an extension of the cell membrane which is covering it. And they're given different names before their structures were discovered. Flagella are long and found usually 1 or 2 per cell, while the cilia are short and often numerous. So this is the first thing. Then you must know this. The projecting from the animal cells and flagella. This you must go through this. Please pause the video here and we go into a little more detail now. Cilia looks something like this, and sometimes you're given a micrograph of this, and you must know what we are talking about when we look at these cilia. Now, this is a diagram which I've taken from the book, the new book. You can see the structure of the cilia. Now, important for you to see the cell surface membrane, which is enclosing it. There's an outer arm and there's an inner arm. And then there are two single microtubules. That's why this is called the 9+2 structure. So 1, 2, 3, 4, 5, 6, 7, 8, and 9. So 9+2, and this is the 2 in the center. So 9+2 structure. And another thing you've got to understand is there's a B microtubule and the A microtubule. And then the A microtubule has these sort of extensions or these arms. And the A has 13. A has 13 protofilaments and B has 10 protofilaments. Well, you can understand why 10, because these ones are 10, and then these ones are all 13. So 13 protofilaments and then 10 protofilaments, and this is called a double microtubule. So this is what you have to doublet microtubule. So these are these doublets, one doublet and two and three and so on. So 9+2 structure. And this is the 9+2 is the structure of the basal body. So we've got to understand what is the basal body. And the basal body is 9 triplets. So 9+2 and 9 triplets, you've got to differentiate between them. Now another diagram from the book, which we have just seen, the Cambridge book. So 9 triplets are part of the basal body. So the 9 triplets are part of the basal body now. So this is inside, this is the cell membrane. So this is inside the cell membrane. This is inside the cell membrane. And this is the one which is on the outer side is the 9+2 structure. So the cilium and the basal body, you've got to know the differences, showing you a few more diagrams on this. So both the cilia and the cilia are just shorter, flagellum are longer. They both have basal body. So basal body is inside. This is the cell membrane, which is being shown here, and then we've shown you a cross section of it. So this is the basal body. This is the section of it shown here. Basal body has 9 triplet, while on the top is the 9+2 structure. So this is the 9+2 structure, and this is the 9 triplet structure. Another micrograph showing this. You might get a micrograph on this, so please be very clear on it, which one we are talking about when we are talking. So 9+2 and 9 triplet, how they are different. Now, as per the new syllabus, you need to know the virus structure a little more. They've added something to it, and they've said, well, usually we used to say just a protein coat, and it's called a DNA or an RNA strand. So it can be the nuclear material can be DNA or it can be RNA. So it's an or, not both. In eukaryotes, of course, we have DNA and RNA. So the virus has a protein coat, which is the capsid contains the virus genetic material, DNA or RNA. Then there are surface proteins, and then there's the virus genetic material. So you can see these labels. And the outer viral envelope is made from phospholipids, and the viral envelope is made from fatty lipid molecules taken from cells in the host. So you have to understand is that viruses would have also some viruses, not all. Then the next thing new in the syllabus is this part 4 of the chapter on enzymes, which I'm just outline the use of a colorimeter. This is a calorimeter, not a calorimeter, for measuring the progress of enzyme catalyzed reactions that involve color change. Now, as you can see here, what is a calorimeter? Using a calorimeter to measure the progress of an enzyme controlled reaction, method is used for measuring the progress in which there is a color change. A colorimeter can be used to measure the color change. This is a colorimeter. Please look at the spelling. I mean, I don't want you to write CALOR. So it's COLOR. And a colorimeter actually measures what? It measures the color of a solution. Like if you got a light blue, dark blue, or a light black or dark black, by measuring the absorption of different wavelengths of light. So basically, it measures the darker the fluid, more the light will be absorbed. If you're going to have just water in front of it, of course, all the light is going to pass through it. Nothing's going to be absorbed in it. So this is what you have to measure in a colorimeter, is the absorption of different wavelengths of light. Now, as you can see here, I've given you this from the page from the book. You have the light, you have a filter, you have a solution in a tube or cuvette, and a light meter and a readout. Now the diagram shows how the colorimeter works. Photograph of a range of colors obtained from the iodine test during the course of an experiment investigating the digestion of starch by amylase. And it is also easier to observe the course of the reaction if you mix starch, iodine, and amylase in a tube and take regular readings of the color of the mixture in this one tube in a colorimeter. However, this is not ideal. However, the iodine interferes with the rate of reaction and slows it down. So this is the way that a colorimeter would be used for a reaction. And of course, you can plot a graph of it, you can plot a curve of it, amount of the starch remaining against time. You can then calculate the initial reaction rate in the same way. And you can do it for catalase, hydrogen peroxide reactions described previously. So you can do these in different ways by seeing the color, how much light is absorbed and how much light can pass through it. So the lighter the color, more light will pass through it and less light will be absorbed. So this is the colorimeter story, which was new in the syllabus. In the chapter on the cell membrane, you have these two which are slightly a little more detailed, a little more, I would say just a little more. It's not such a lot of a change in it. This is actually the same, but look at the wordings. Describe the role of phospholipids, cholesterol, glycolipids, proteins, and glycoproteins in cell membrane with reference to stability, fluidity, permeability, and transport. Transport is carrier proteins and channel proteins. Now they're specifically being mentioned. Cell signaling and cell recognition, which is cell surface antigens. Then this is a little more detailed now. Outline the main stages of cell signaling leading to specific responses. Number one, secretion of specific chemicals, and these chemicals have been named ligands. So you must know these ligands from cells, and then transport of ligand to the target cells, and then the binding of the ligands to the cell surface receptor. So there would be a cell surface receptor on the target cell. Like I always ask you, this question is, how does insulin know it has to go to the liver cell? Why? Because the liver cell would have some sort of a receptor on its surface, which where the insulin molecule will just fit into it. So cell surface receptors, ligands, chemicals, and transport of ligands to the target cells. Naturally, those transports of ligands, how they would be transported to the target cells, and then binding of the ligands to the cell receptor. So this is a little new. I want you to know these names, and I want you to be clear on this. This is a diagram showing you the signal, which is a ligand coming here, and you can see how this is coming here and binding to a surface receptor, which is a protein in the cell membrane. Then there's a G protein, which is activated, which then further activates another enzyme, which makes a second messenger, and then the activation and the second messenger, and then signaling cascade with amplification at each step, and then activated enzymes and other activated enzyme, and this results in secretion or transcription or movement or a metabolic change. Basically, it's the chemical which is going to be transported maybe through the blood, and then it reaches the receptor where of course it's going to start working, activates the G protein, that G protein, of course, and this whole thing, it spans the whole membrane, and this of course results in a change in the shape of the receptor, and then that activates the G protein. So change in the shape, transduction, and then that is going to result in activation of the enzyme which is going to produce the second messenger. So please pause the video here and have a look at this, and then of course we will go on to the next part of the updates. This chapter has the biggest chunk which is new in the syllabus. So please have a very clear idea on this because this would not be in the old book. So what is new in this syllabus is part 3, which is new. This whole thing is new. Then part 4 is new. This is all new in the syllabus. Please understand this. This is all new in the syllabus. So the rest of it is all mostly the same, but then we need to be talking about this, which I'm going to talk in detail about the part 3 and part 4. Then in protein synthesis, this is new. Describe the part 2 is new. You must be very clear on this. Then 4, 5, 6, and 7 is all new. This is all new in the syllabus. So I'll be start discussing this and then we'll go on to further explanations. Now, the first and the most important thing is the 5' end and the 3' end. So you see, these are the 5 end and the 3 end. You can see here, this is written here 5 end and this is written here 3 end. And then this is the 5 end and then this is the 3 end. So how do you figure this out? You've got to figure this out. You see, this 5 is at the top here, 5 end. And here we have 3 here. This is a 3 here at this end. So this is, I mean, this is like this. This is, and this is facing upwards. So you've got to be understanding is that, you know, how the 5 end and the 3 end, and then the hydrogen bonds between the, thymine and adenine, there are 2 hydrogen bonds, and between cytosine and guanine, there are 3 hydrogen bonds. And I always tell you to remember it. C is the third letter of the alphabet, A, B, C, third letter of the alphabet, so 3 hydrogen bonds. So deoxyribose 5 end, which in the 5 carbon is at the top and the phosphate is at the top, and here we have the 3 end. So 5 end and the 3 end, everybody must know this. And this is because this is anti-parallel. So you can see how DNA polymerase can only work in 5'3" direction, while in this situation it will work in this direction. So we're going to have a problem in DNA replication because DNA polymerase can only work in this direction. So one is like this and the other one is like this. So this is anti-parallel. The strands are anti-parallel, but the DNA polymerase also has an issue how to work this out. So this is the first thing that you've got to know. Then the second thing you've got to know is another diagram, maybe better explanatory, is the 5 end and the 3 end. So this is the 5 end, this is the 3 end, then this is the 3 end, and this is the 5 end. And then you see this is the 5 here, and 3 here. And so similarly here you have the 5 here and the 3 here. So you've got to figure this out. You've got to see this one 5. So it's going to go like this, it's going to go like this. So this is the 3 end. And then we've got, let me use another color. So you've got the 5 end which is going here, this. So you've got to really figure it out. They give you this in the MCQ, or you've had it if it comes in the MCQ. First you've got to understand is the phosphodiester bond. And how the phosphodiester bond, as the syllabus says, is the linking of nucleotides by phosphodiester bond. So you've got to see the 5 end phosphate attaches to the 3 end of the upper nucleotide, and that results in a phosphodiester bond. Please pause the video here and have a look at this. Then you look at this here, and this gives a more detailed version of it. So this is the 3 end, which we've got to understand. This is the 3 end and this is the 5 end. So between the 5 carbon and the 3 carbon of the upper one, it forms the phosphate, links with it, and a water molecule exits, and the phosphodiester bond is formed. So diester, phosphodiester. So the diester bond is the one which is going to join 2 nucleotides, and 2 phosphates, as you can see here, have been released. So 2 phosphates, because this was a deoxyribonucleotide triphosphate. So this is the triphosphate, which was actually is the activated nucleotide, which are brought here, and then of course the 2 phosphates exit from it. This is called a DNTP. You'll study this in A2 as well. DNTPs are what? Deoxyribonucleotide triphosphate. So these are called DNTPs, deoxyribonucleotide triphosphate. Now, coming to the semi-conservative DNA replication, you've got to understand is that the 5'3" direction. So if this is the 5 end, I'm going to use another color. This is the 5 end. So then this is going to be the 3 end. Now the leading strand starts from here. So the leading strand is going to be in this direction, which is the 5'3" direction, and it's going to be synthesized continuously because this is going to continue like that. But when it reaches here, you see we have a problem. This one has the 5 end is here. So if you look at this one, which I'm going to give another color, this is the 3 and then this is the 5 end. Now the 5 end, that means this has to be somehow made here like this. Oh, we can't make it like this. So how do we then replicate it? So it is a discontinuous replication and it is done in tiny fragments, which is this way, this small fragment here, small fragment here, small fragment here, because this would be the 5-3 direction, because this would be the new strand which is going to be made, and this would be the 5-3. So this would be the 5 and this would be the 3 direction. So then we would have this in a continuous manner. And why is this called discontinuous? You've got to understand that, because when this opens up more, you see, this had opened up only here. But as this part also now opens up, so now we have it like this. So this was being made like this. So here we have this strand made like this. Now another one will be made like this, while this one will continue. So the leading strand will continue. The leading strand will continue. This is the leading strand. The leading strand will continue. This is the leading strand. But the lagging strand will be made in short pieces, like this piece will be made, then another piece will be made. So this piece will be made, and then another piece will be made, and there's a slight gap in it because it's not continuous. And that is why this is called discontinuous. The lagging strand is discontinuously made. Now, looking at the different steps, we've got number 1, helicase unwind, the parental double helix. Number 2, single-stranded binding protein. Now, you look at these little red structures. Now these are called the single-stranded binding proteins, stabilize the unbound parental DNA, because you see the hydrogen bonds between the bases are broken. Now we need to help them to keep them separate so they don't join up together again and form the double helix again. So single-stranded binding proteins stabilize the unbound parental DNA. Number 3, the leading strand is synthesized continuously. I told you 3 and 5. So leading strand is synthesized continuously in the 5-3 direction by DNA polymerase. The enzyme DNA polymerase is going to do that. Number 4, the lagging strand is synthesized discontinuously. The primase synthesizes an RNA primer. So you can see these RNA primers. These are the RNA primers which I'm going to mark in red. These are the RNA primers because you see DNA polymerase cannot pick it up straight away. So RNA primers, which is extended by the DNA polymerase, and it just forms an Okazaki fragment. So this whole structure, the RNA primer plus the newly synthesized DNA, is called an Okazaki fragment. So number 5, after the RNA primer is replaced by DNA, then we remove these RNA primers and we replace by DNA. That's another DNA polymerase is used, and then the DNA ligase is going to join it. So we're going to have the DNA ligase then joining these pieces of DNA together. So these pieces of DNA are going to join. So it's going to form the phosphodiester bond and is going to continue it. So lagging strand and leading strand. So this is of course what we have got to be very clear about when we are. So the lagging strand was the number 4 here, which we are talking about, this one, which is the lagging strand. Another diagram showing you the leading strand and the lagging strand, parental DNA. You got to remember it is the 3 end, which is going to be the one which is going to decide which is going to be the leading strand. I can think of MCQs being based on this syllabus point. So at leading strand and the lagging strand, template strand. Now remember, this is the template strand, the original DNA. So this is the template strand and this is the template strand. So the one in dark blue is the template strand. And then you can see the enzyme DNA ligase joining the Okazaki fragments together. So you've got the Okazaki fragments here, and then the joining them together is the DNA ligase. So I'm sure they're going to set you MCQs on this, which is going to, so of course, and you can see how the replication fork keeps on extending. You see it was here, now it is here, so it keeps on extending. And then we would have another Okazaki fragment here being made. So more Okazaki fragments, and then of course joined by DNA ligase. Now another diagram to show you the origin of replication and how it is in both directions. You've got to understand this. You see this bubble forms, this replication bubble, and then the leading strand. So the leading strand is the origin of replication. You can see here I've drawn it in a number of ways to show you this. So this would be this is the leading strand, and then the red would be the lagging strand in this situation. But in this situation, then this is the leading strand. in this direction, because it will be 5 end here. So this will be, of course, the 3, and you see this will be the 5 end. So the leading strand is going to be made in this way, because this is the 5 end, and this is going to be in this direction. So 5, 3, I might have said it the wrong way in the beginning. And then because this is going to be now the 5, 3, so then it is going to be in the other way. So it's going to be in the pieces, which is the Okazaki fragment. You can see this here, lagging strand, leading strand. Here the lagging strand is this one and the leading strand is this one. So please pause the video here, have a look at it. There's more details and the RNA primer being added to it, and then the DNA polymerase and the parental DNA. So please have a look at it. I will continue this in another video in which some portions of the syllabus updates will still be remaining. And I've completed this part of it, which is new in the syllabus, and the leading and the lagging strand, and the continuous and the discontinuous, which we had to do in this new syllabus. And thank you, and please, another video will follow very soon.